Our next presentation is Umoja Biopharma, and I'd like to welcome David Fontana, Chief Operating Officer and Chief Business Officer, to the stage.
Well, good afternoon, everyone. Lovely to be up here. My name is David Fontana. I'm at Umoja Biopharma, and we're the leading in vivo CAR-T company. And on behalf of the ARM, I want to thank them for arranging a beautiful meeting. Hopefully everyone's having a blast. I'm sure having fun here. You couldn't beat the beautiful setting, and thank all of you for making time for me today. I have forward-looking statements, so I basically, what I can say to you is I'm going to say a lot of gonna do's and wanna do's, but you should probably take those with the appropriate risk statements. I'm gonna start the presentation showing this vial, and this actually means a lot to us at Umoja. It's from our first GMP manufacturing facility. So 2 years ago, I was here saying we're gonna build a facility and manufacture out of Boulder, Colorado, and sure enough, here's our first vial, coming out of theirs, and this will be a vial in one of our clinical trials this year. But more importantly, I want to talk about the potential of what's in that vial, and that'll be my presentation today. So our VivaVec particles, we have the ability to deliver these small particles, proprietary particles, directly into patients and basically generate CAR-Ts into patients. So for example, a CD19 CAR, by giving our first product, VV111, we can generate very nice PK profiles in the CAR-Ts. We also see the corresponding biology. For example, if you look at tumor killing or B-cell aplasia with these VivaVec particles, again, no cells involved, we see the appropriate biology of B-cell aplasia and tumor killing. What's important, though, if you think about the potential of this, is this will be sitting on a shelf in Hackensack, New Jersey, and if you want to get treated in Hackensack, New Jersey, you don't have to go to Memorial Sloan Kettering. You can be treated right in the local Hackensack Oncology. Vice versa, if you're in Memorial Sloan Kettering, by all means, can get treated there. But I think that's a big problem with access now, and we're trying to solve these problems with the VivaVec product. Secondly, the cost of these things. These are going to cost, and I'll show you a slide later in the deck, these are roughly going to cost the same as monoclonal antibodies by the time we get done. And we're quite excited about that. So what we're hoping with this potential of this vial is off-the-shelf, activity very similar or better than the CAR-T, a safety profile maybe even a little bit better, and eliminating all the access problems we see with the currently ex vivo CAR-Ts. Wonderful drugs, but there's access problems. And hopefully I can convince you we're on our way to that today. So we're in a great position at Umoja. We have a robust pipeline. We're starting with 2 clinical trials that are initiating and actually open for enrollment at this time. One's a VV111, it's a CD19 phase 1 clinical trial open up in the United States and Australia, and the second one, a VV400, which is CD22, which is opening up in China at an IIT this year. We're gating the rest of our pipeline on getting activity or proof of concept out of those molecules. Reason being just we're a small company and we want to gate our finances, but we have a robust set of relationships to build our pipeline with lots of potential behind there. If I focus on the middle a bit, and this is kind of our what we think is our game changer, we eliminate a lot of the limitations of the CAR-Ts. Again, as mentioned, it's off the shelf. There's no lymphodepletion required, and I'll show you a slide. We don't have lymphodepletion, it's not required. And what we do have, though, is our own artificial cytokine system that can actually deliver the growth signals right to those engineered cells and only those engineered cells to improve persistence in that one. We own our own supply chain, again in Boulder, Colorado, a beautiful facility. We have capacity to make up to 13 or 14 200-liter GMP batches. It looks like now one GMP batch can cover a phase 1 study, so we've clearly got great capacity to go forward. And our data so far looks like we're going to at least meet or exceed what we see with the ex vivo CARs in non-human primates, and I'll show you some of that data in a bit. Financially in good shape, $300 million raised to date. We're actually going to close a Series C by the end of the year, so that's coming up, good news. We're very fortunate to we have 2 relationships with AbbVie. They've been a wonderful partner, very generous, and working very well together, and that's really helping pay the bills. And what this capital efficiency model means really that, through our partnerships, disclosed and undisclosed, our CapEx or our OpEx for manufacturing a lot of our platform is really covered by those relationships. And there's a lot of milestones coming up. So the money we get from investors is really going to build an internal pipeline. And finally, I want to make the point that we're taking a strategy of doing lower cost trials by working with our partner ISO, who is the leader in CAR T in China. We're working with them. They have a lot of experience in running clinical IITs, and we're teaming up with them to run our first trials here. It's more cost effective and time effective, and we'll get data out of those trials hopefully a bit faster than we would out of a US IND. So let me talk about the business engine. So this is our VivoVec. VivoVec's our proprietary product here. So it's the third generation lentiviral vector product. And for lentiviral vector products to work, they need a couple things. First of all, they need a way to bind, right? They need a way to attach to the T cell. So that's done by our CD3. We have a CD3 binder that binds to the T cell and activates the T cell. Beyond that, though, we did a lot of engineering around the lentiviral particles, which is really our specialty. The surface engineering is our specialty. We were able to add additional co-stims and tropism agents to drive both greater activation, greater tropism, and better binding of those particles. So once it binds, it starts the cascade of activation and starts the cascade of priming. And then you need to deliver those transgenes, the payloads that are in those VivoVec particles. These particles contain all of the genetic information, the payloads that we want to deliver to the T cells and transduce them there. And so that's done through our CoCal mechanism. So the CoCal delivers those efficiently into the T cells, and through the standard lentiviral vector machinery, transduces the cells. And those who know about lentiviral vectors, it's not only the parent cells that get transduced, it's the daughter cells as well. So this is a permanent integration. So the initial cells we engineer and all the follow-on cells will carry, for example, the CD19 CAR with those. In this example here, you can see that there's just a CAR, but we also have in the first two products we're testing the CAR, and then that synthetic cytokine system I'm not going to talk a lot about because not enough time in this presentation. So we're delivering two payloads to these with the VivoVec particles in a timely way. So what makes us different, we feel? So I've talked a bit about what's different than ex vivo CAR. We again, no lymphodepletion, and we will eliminate a lot of the access issues. No apheresis, we don't touch cells, we don't ship stuff across the country. It'll be sitting there from Colorado right to your doorstep, kind of thing like that. Versus allogeneic CAR Ts, again, persistence, we have no trouble with persistence. We've done plenty of studies, and I'll show you some data. And again, we removed the lymphodepletion, so really excited about that as well. I really like T-cell engagers. I think they're making a great play. But two issues I see with them. They still have this toxicities and the repeat administration, right? So you've got to repeat and come back and forth. I think one of the beauties of what's out there now, the CARs, the Carvyktis, and the Breyanzis, is really that ability of one and done that patients quite like. So T-cells engagers, and I'm unaware of any data on a T-cell engager that matches a CAR T in the same target. So I think the CAR Ts will, at least until proven otherwise, hold efficacy advantages over the T-cell engagers. And then finally, we're not the only ones in the space. There's a couple folks in this room working in the space and very competent and great companies. We think we have differentiated IP with our multi-domain fusion protein. We really have done a lot of work. What I told you about is the co-stims of trying to match the T-cell antigen-presented cell interactions. And we think we've tightly done that, and some of the data is there. And then finally, we think we have the best non-human primate data. We're all using the same kind of CD20 non-human primate data in healthy monkeys to look at efficacy and PK, and I'll show you some of that data now. So this is our molecule called VV310. It's a molecule that has CD20 but does not have the cytokine system in there. And so in the black is a study done, the animals done by Dr. Mike Jensen at Seattle Children's Hospital. This is his CD20 with an ex vivo autologous CAR. They lymphodeplete, and you can see on the left-hand side, after giving the drug at day 0, you see a really nice B-cell depletion, right, maxing out at about day 7 and lasting about 30 to 35 days. Correspondingly, on the right-hand side, you see a typical peak, as one would see in plasma levels of CAR T, a nice peak that is around day 7. So very typical what you'd see in the clinic, and so a pretty good model. The VivoVec stuff's shown in orange, and you can see that our administration of our VivoVec product, we see a corresponding equal decrease in B cells, so B-cell aplasia, lasting a little bit longer than what we see with ex vivo CAR Ts. Again, we're not lymphodepleting, just giving VivoVec particles straight. And you can see a beautiful PK profile, very consistent and dose-dependent, as we'll show later, dose-dependent increase in CAR-Ts and response in the blood. So really fantastic data, which gives a lot of confidence to us going into the clinic if these results would mimic what we see in the clinic. Our pipeline, so we're taking very low risk in our targets, and our primary target is CD22. We love that target, and I know others in the room, including some of my former colleagues, like that target as well. We like it for lots of reasons. I'll touch upon that a little bit later. We're starting a study this year, and we'll be dosing patients by the end of the year and for certain by the beginning of next year. We're also taking that drug 310 that I've just shown all the monkeys on into autoimmune disease early next year as well. We've got a great package coming, and hopefully at ASH, at ASH we have an oral presentation on this new data set that includes not these monkeys shown here, but also a full IV and intranodal dose response curve. So that'll be quite interesting for everybody. And what we've done now, as I mentioned, we're gating our portfolio spend on waiting till we get some proof of concept in the clinic, but we have relationships with IOSO and a company called Nona Biosciences, so we're building and screening binders to look at multiple myeloma targets like BCMA, GPRC5D and such, and also several solid tumor targets, so our portfolio is ready to go when we get clinical proof of concept. And I wanted to mention our partnership with AbbVie. So we have 3 ongoing trials. We have 2 partnerships with AbbVie, a development partnership, which includes the VV111 for we're starting in non-human primates, NHL and CLL. So that trial is actually open for enrollment right now. It's in its partnership with AbbVie. And we have 2 undisclosed programs. One's in hematology, a very novel hematological target, and then solid tumor targets as well. So again, AbbVie has been great to work with. We get a lot of milestones, a lot of synergies by working together with a big pharma that contributes to our development of these programs. Quick clinical overview. I mentioned the CD22, so our molecule is called VV400. We're starting in IIT in China with IOSO, and what we're really looking at is patients post CD19. So a lot of patients fail CD19 after they receive the CARs, CD19 CARs. And what's been shown by Stanford and some of the colleagues in the room at Cargo, that CD22 responses are maintained in those patients that fail CD19. So we're basically mimicking those trials, and we're going to go in pretty aggressively with IV dosing and IV route of administration, along with using our rapamycin, which is our cytokine activation system. Again, I mentioned we like CD22 for the following reasons. First of all, only about 20% of the people getting CARs right now, or only 20% of the patients eligible for CARs right now are getting the ex vivo autologous CARs. So we think VivoVec can really help maintain that one. I mentioned CD22 relapses. 60% of those who get responses relapse in those patients. So we see some advantages there as well. And then there's opportunities in frontline and other lines of therapy with VivoVec and CD22. Again, an unexplored target. And I can't wait to the day where maybe it's us or Cargo will explore CD22 in autoimmune disease as well. There's no reason to believe it wouldn't work. Our Invicta trials, our CD19 trial, what's different about this one is we're doing a lot of work on route of administration and dosing. So we're going both intranodally, which is a lot of our data. T cells get generated, and the manufacturing system for T cells is in the nodes and lymph system. We're going directly there, but also doing IV, so we'll get a good comparison of how to dose going forward. Won't spend much time on this slide, but I just did want to say that we see our opportunity pretty big. We're going to start small with our CD19 failures with our CD20 CAR, and then move up lines of therapy. The data that is shown by Stanford is really compelling that there might be disease states and there might be a safety advantage of CD22 in non-Hodgkin's lymphomas in particular, and who knows what to see. But also looking forward to our pipeline, looking at different molecules in solid tumors and other hematology indications as we go. So our manufacturing facility is absolutely gorgeous. It's called The Climb. We try to name all of our buildings. So we've got The Climb in Colorado and the Ski in Seattle, and someday the Surf down in San Diego. So we're looking forward to that. But it's a state-of-the-art, built from scratch manufacturing facility. What we really like about it is we control supply chain all the way through it. So little companies, we've already switched priorities 3 or 4 times. We no effect. If you're working with a CDMO, you can imagine the impact of that. So we really like that. But I wanted to highlight this. Look at the cost of goods. Our initial phase one, go to clinic, we had a certain benchmark, and we've got that benchmark out there, knowing what it costs. But we've put together a team called Optiforce, who's really spent a year now optimizing, pulling the levers for manufacturing and optimizing it now. We've got up to 20-fold improvements already in manufacturing yield and productivity. Our estimated cost is going to be $6,000 to $8,000 versus the $250 for the ex vivo cars. And I want to close on this. I really want to first of all
Thank you, Mr. Fontana. It's time for our next presenter.
All right, thank you. Says it all. Thank you, guys. Cheers.